Hello, my name is Anna Beck from Laurel School, and my project is the effect of natural killer cells on neuroendocrine tumors with Dr. David Wald from the Department of Pathology at the Case School of Medicine. We are working with natural killer cells, which are lymphocytes, or a type of white blood cell, that is cytotoxic, which means that it is capable of destroying virally infected or cancerous cells in the human body. So part of the immune system. And they operate according to the activating and inhibitory receptors on the cell surface, which detect the other receptors on other cells' surfaces to see if they are infected, or NK cells will also operate according to cytokines, which will cause them to proliferate or become active and start killing. So immunotherapy is fighting cancer by aiding the immune system. And in the case of NK cells, this means removing the patient's immune cells, expanding and or modifying them, and then re-injecting them so that those immune cells can fight the cancer with new abilities or heightened abilities. And we are investigating NK cells in this experiment because T cells, which are usually used for immunotherapy in this way, can cause side effects when re-injected, and we're experimenting to see if NK cells, which probably don't have those side effects, are effective against the same types of cancer. So the cancer that we will be looking at in this experiment is neuroendocrine tumors, which, according to this diagram, can be found throughout the neuroendocrine system. And they are very difficult to diagnose, so they are often not detected until the cancer has progressed to a stage where it can no longer be treated, and that is often the case with pancreatic cancer, and therefore pancreatic cancer is fatal in most cases. So the goal of this experiment is to see if NK cell therapy can be a treatment option for pancreatic cancer through an experiment called a killing assay. So to prepare for our killing assay, we first need to grow our cells. To grow our NK cells, we will first isolate them from the blood of a healthy donor using a substance called Ficol that will divide the blood into layers. And then from the peripheral blood mononuclear cell layer, we will remove the NK cells. And then we will grow our neuroendocrine tumor cells from a QTP1 pancreatic islet carcinoma cell line. For the killing assay, we will first stain the cancer cells with calcium, which will allow them to be counted in the flow cytometer, and then we will fill wells of our 96 well plate with different ratios of NK cells to cancer cells to see which ratio is most effective for killing the cancer cells. And then we got our results from the flow cytometer, which counted up the numbers of live cells. And as you can see from the rightmost two columns, as the ratio of NK cells to target cells increased, the total count of live cells did decrease, but not very much, considering the ratios of NK cells that were present. You see that the very last column in each experiment, A, B, and C, is 10 to 1, yet the number did not decrease by that much of live tumor cells. And so this data implies that NK cells are not effective at killing these neuroendocrine tumor cells. And our options for future experimentation are to either try a different cell line of neuroendocrine tumors, because it's possible that this particular cell line was not responsive to NK cells. Or it is also possible that NK cells cannot be used as a treatment at all against neuroendocrine tumors. They're just not responsive. And CAR T cells, shown in the picture below, might be just a better option for immunotherapy against neuroendocrine tumors. I'd like to thank Dr. Wald for supervising this experiment, Kalpana Gupta for helping to run this experiment, David Bush for helping with the writing, and Dr. Berger for running this program. Thank you.